Hello and welcome to today's webinar. My name is Courtney Akitaki. I am the Light Sheet Application Specialist and Marketing Manager for ZEISS Microscopy in North America, and I will be the moderator for this webinar. While people are dialing in, let me quickly run over a few technical points. The webinar will take approximately 45 minutes, after which we will have about 10 minutes to answer some of your questions. In case you have any technical issues, please use the chat function in your GoToWebinar dashboard to send a message and we will try to help you. And if you do have any questions, please use the questions function in your GoToWebinar dashboard. We will collect all questions and save them for later in the Q&A session at the end. I'll now introduce today's speaker, Dr. Sunil Gandhi from the University of California in Irvine. Dr. Gandhi's research is focused on understanding plasticity of neuronal connections in the young brain and developing technology to reactivate plasticity in older brains, which would impact therapies for brain repair after injury, stroke, neurodegenerative disease. Dr. Gandhi has pioneered the use of embryonic inhibitory neuron transplantation to rewire the adult visual system, and more recently, his group has developed new methods for whole brain visualization using new brain clearing methods and light sheet microscopy. Today, he will be telling us about brain-wide quantification of inflammation and neural activity at single cell resolution. And with that, I'll turn it over to Dr. Gandhi.
Thank you so much, Courtney, for that introduction. Thank you all for attending this webinar. As Courtney said, I'm a professor at the University of California, Irvine. I should also mention that I am the co-founder for a company, Translucence Biosystems, that's funded by the NIH Brain Initiative to disseminate the technology to other neuroscientists that I'll be discussing in this talk today. So, as Courtney mentioned, I'm a visual neuroscientist. I'm concerned with the circuitry of the visual system and its underlying anatomy. And in the last few years, the advent of new technologies in brain clearing, light sheet imaging, and computational image analysis has really transformed our approach to neuroanatomy. And it's enabled us to ask new questions, particularly looking at the brain-wide quantification of cellular features in the rodent brain. So that's enabled us to ask questions that are novel and that are far more quantitative and rigorous than what was previously possible. And so my main message in this talk is that these new technologies are really going to change how we do neuroanatomy. So what do we mean by brain clearing? I'm going to show you here a video of a mouse brain as it's cleared using the iDisco method for tissue clearing. This is sped up, but what you'll see is that the opaque tissue becomes transparent in this process. So this technology really changes how we can approach neuroanatomy. It lets us go beyond just the first few millimeters of tissue and really look across the whole brain using fluorescence microscopy and to ask questions about its organization. Now, there are a number of techniques that have emerged in recent years for brain clearing and tissue clearing in general. A few of those methods are identified here. The method iDisco, developed in Marc Tessier-Levine's lab, is the primary method that I'm going to talk about today. Of course, many people are familiar with hydrogel-based methods and other organic solvent-based methods. All of these different methods have their advantages and disadvantages. I'm not going to be talking today about these differences. I'm instead going to focus on how imaging and image analysis lets us ask very interesting questions about how the brain is organized. So, as an anatomist, I have looked at the brain using thin section histology, as we have over time, by chopping up the brain into thin sections and laying it out on slides, then trying to use atlases to locate exactly where in the brain we're looking. Now, this whole enterprise is fraught. It's quite labor intensive. It can be difficult to reconstruct the whole brain perspective. So a lot of three-dimensional information is lost. But with the advent of tissue clearing, it's now possible to hold the brain intact. And here you can see a tube with a mouse brain before and after clearing. Before clearing, it's opaque, but then after clearing, it's pretty much transparent. So now we can, instead of using a blade to chop up the tissue, we can instead use light to optically interrogate the brain wherever we would care to look inside the tissue. To do that, we need to turn to a different type of microscopy, and that's light sheet microscopy. In standard epifluorescent illumination shown on the left, we excite a size, in the case of whole brains, we would excite the tissue, even though it's optically clear, we would end up damaging the tissue above and below the focal plane. So this type of illumination isn't appropriate for whole brain imaging. Instead, we can use an old technique of illumination along the orthogonal axis using a light sheet to illuminate just at the focal plane of the detection optics, sparing the out of focus plane from any photobleaching. So this method of light sheet microscopy combined with whole brain clearing lets us image very large sections of brain tissue all at once and at very high detection efficiencies. So recently we teamed up with ZEISS Microscopy to retrofit one of the existing light sheet microscopes from ZEISS, the ZEISS C1, to adapt it for use in brain clearing. Originally, this system has widely been used for visualizing small samples such as zebrafish or small embryos. But by using this new adapted system, we can look at larger samples such as whole rodent brains. We also can accommodate the optics to work for the high refractive index solutions that are needed in order to examine cleared tissues. And so what this system lets us do is tap into the superior optics of the ZEISS light sheet system, but to speed up the acquisition times and improve the imaging of very large samples. So I'll show you an example in this next slide of a mouse brain that is in which the neurons are sparsely labeled with GFP. This is the Thai-1 GFP mouse line that we cleared, stained for GFP using the iDisco protocol, and then imaged using this modified ZEISS system in around 25 minutes. And here you can see the brain, the neurons, the cell bodies of the neurons you can see as these spots. You can see the dendrites and then axons coursing through the circuitry. Here you'll see a zoom in on the motor cortex and then the individual axons of the descending corticospinal tract. So this video demonstrates the power of this technology, of course, for visualizing the complex circuitry of the three-dimensional brain. It's quite exciting and illuminating. This video won the video contest at the 2020 NIH Brain Initiative Investigators meeting. But apart from making beautiful videos, what I think brain clearing and light sheet imaging can really provide the field is a new way to get very quantitative and rigorous about brain-wide patterns of cellular features. So in my talk today, what I want to do is take you through 3 or 4 examples where we have tapped into this potential for this new technology to enable us to quantify patterns of neuroinflammation and neural activity that would have been nearly impossible with thin section conventional histology. So in this first application, I'm going to tell you about a study we did together with my collaborator here at University of California, Irvine, Melissa Ledoen, and her lab. Her lab studies the parasite toxoplasma, and it's targeting to an infection of the brain. And we had set out to understand how toxoplasma might change the brain of mice. It's widely known that toxoplasma is takes advantage of the mouse as a vehicle for its reproductive cycle by altering the behavior of the mice such that they become vulnerable to predation by felines. And then it's in the gut of the felines that toxoplasma can sexually reproduce. What Melissa's lab had discovered was that at the time when there is a big signal for the infection in terms of circulating immune cells in the blood, there was also a sudden infiltration of monocytes from the blood into the brain. And we wanted to know whether this infiltration of monocytes might be regionally specific. Perhaps certain circuits were spared and certain circuits were affected, and this might help us understand some of the behavioral impairment that people have documented in mice during toxoplasma infection. So we turned to brain clearing to help us look for those patterns of neuroinflammation. So what I'm showing you here is a section of a toxoplasma infected mouse brain, and we've used a genetic strategy to label the monocytes that infiltrate into the brain. And you can see this in the zoom in over here of the fluorescence image that the monocytes appear as these pill-like structures in the tissue. We use machine learning enabled algorithms to segment out these very large raw image data sets to produce 3D masks for each and every one of these cells so that we can quantify their location, their shape, their relative distribution, and co-localization with other markers. And here's where I think, again, I want to exemplify the power of this technology, where not only can we quantify these properties, but we can really determine their regional focus. So what we do is we take the autofluorescence from the brain samples to align them to the Allen Reference Atlas. And what this lets us do is import the annotations from the Allen Reference Atlas in order to mark the boundaries between areas in each of the different brain samples and precisely quantify where, in this case, the monocytes have landed. And you can see the mean heat map for the density of monocytes depicted here. There are certain regions in which there appears to be more monocytes than in others. And indeed, we can observe in these morphed combined tissues that the prevalence of monocytes seems to both be more intense in some regions, as you can see here. This is a region that I'll be showing you more about, the olfactory tubercle, and less intense in other regions, such as the CA field of the hippocampus. And this is in contrast to another neuroinfective agent sorry, not neuroinfective agent, but a inflammatory agent, LPS, which stimulates a broad-based infiltration of monocytes into the brain, just like T. Gandhi infection. There's a very different pattern, as you can see here, in the brain-wide infiltration of monocytes. So we developed our software pipeline to give us the ability to quantify by brain region how many monocytes were infiltrating. And here you can see a few regions which popped up as having greater than chance preponderance of monocytes. And the gray bar here indicates the shuffle corrected estimate for by chance densities. You can see that most areas actually fall below the by chance levels, suggesting that some regions are not vulnerable to the monocyte infiltration, where other regions aren't. And we found this pretty interesting and surprising. What's shown over here is the data plotted as heat maps. So we had analyzed the monocyte infiltration for a number of different brains, and each column here shows you the density of monocytes across hundreds of different brain areas in the case of the toxo-infected mice. And what you can see is there's kind of a sporadic pattern with some form of consistency, and that this data looks very much like gene expression data that you might get from RNA sequencing. So we adapted some methods from RNA sequencing to apply to this case to try to get a better statistically grounded sense of what areas really were attracting more monocytes in the toxo-infected animals. So what we did was to compare the areas that had monocyte infiltration in LPS versus the T. Gandhi infected condition. And here you can see a volcano plot showing you that differential expression. And in the LPS condition, you see that there is many areas for which there is a greater preponderance for monocyte infiltration, because LPS induces a brain-wide, more uniform pattern of neuroinflammation. In contrast, with T. Gandhi, there are only a handful of areas for which there is a significant degree of selective density or increased density of monocytes. And the olfactory tubercle turns up right here, just like we had seen just in the shuffle corrected estimate for statistical significance. So the takeaway here is that this form of analysis, of brain-wide regional analysis, can detect patterns of neuroinflammation that we might not have been able to detect with thin section histology, and that we can use some of the statistical frameworks from RNA sequencing to allow us to make many different comparisons statistically rigorously, and use this as a jumping off point for then pursuing the hypothesis that perhaps the olfactory tubercle is selectively vulnerable to monocyte infiltration in toxoplasma infection. And you know, this is an interesting and appealing hypothesis because the olfactory tubercle is located centrally between the ventral striatum and the olfactory nucleus, the olfactory cortex, and therefore it's well situated to integrate different signals from different sensory modalities to shape the animal's motivated behavior. And so its selective vulnerability in toxoplasma infection may explain why mice become vulnerable to predation. Okay, so let me now transition and tell you about a second application that we have made of brain clearing technology. And in this case, we're going to move from neuroinflammation to neural activity. So when neurons are active, there is a concomitant flux of calcium ions into cells that we know activate a series of what are known as immediate early genes, such as CFOS and ARC and others, and that this concept has been used extensively to visualize the neurons that are selectively active during a particular experience. Now, the traditional IEGs can also be activated by other paracrine signaling systems, muddying the interpretation that we can make about the patterns of IEG expression and their connection to recent neural activity. In contrast, the immediate early gene NPAS4 was identified by Greenberg and colleagues to be much more selectively tied to neural activity, making it in principle a superior marker for recent neural activity during an experience. But it hasn't been used nearly as extensively to mark anatomically the cells that were active in a recent experience. So together with Translucence, we have been developing a pipeline for mapping neural activity using NPAS4 together with other IEGs. And this is what the pipeline looks like. So the animal first goes through an experimental manipulation. Perhaps that's a behavioral one, or perhaps it is some neuroinfection or a genetic model for some disease or condition. Perhaps it's to test a drug. Once the animal goes through this experience, we harvest the brain, then clear it and stain it using the iDisco protocol, image it on the mesoscale imaging system in a ZEISS light sheet microscope, and then we can detect brain-wide patterns of the expression of these immediate early genes, NPAS4 and CFOS, and then use our computational pipeline to determine statistically distinct patterns of neural activity across hundreds of brain areas. Okay, so the simplest kind of experience that we could think of to look at in this brain-wide way is to take an animal that is sitting housed in the dark and then bring it into the light to then see what brain areas become activated. We of course expect the visual system to become activated. We also would expect that circuits that get activated as the animal is exploring the lit area of the box might also become activated. So this is what the light sheet data looks like. This is the whole brain perspective and zoom ins on visual cortex with NPAS4 staining in the top row and CFOS staining in the bottom row. And what you can see is that in visual cortex, there are cells which can be marked with NPAS when the animal has gone from the dark into the light. Same thing with CFOS. And then we use our machine learning enabled algorithms to mask out each and every one of these NPAS expressing cells or CFOS expressing cells. Then we can align these brains, again, using the autofluorescence signal to the Allen reference atlas so that we can quantify where the cells are residing that are expressing NPAS or CFOS and determine whether these patterns are statistically significant. So what's shown here is a number of brain areas for which there was a clear increase in expression of NPAS as a function of time. So the way to read this graph is that these are samples that are taking with increasing lag from the time that the animal was exposed to light, from 0 to 120 minutes. And each column shows you the density of NPAS expressing cells in a bunch of different brain areas. So you can see that initially there's no expression, but then it starts to come on. And this gives you a sense again of the kind of natural variation that we find in this data. So here is a density plot for different brains in which the animal had been sacrificed with increasing delay after the end of the light exposure. And what you can see is that when we quantify the NPAS density by region, we can find that the primary visual areas are expressing NPAS maximally with a 60-minute lag to the onset of the experience. Retrosplenial cortex also is activated, the hippocampal regions, as we might expect. And when we look at the differential expression, comparing the 60-minute time point to the 0-minute time point, you can see here in this volcano plot the significant areas that are expressing NPAS. So this is a framework then for allowing us to explore and identify circuits that may be activated upon this new experience. And there are a number of surprising and interesting areas embedded in this data set that then we can generate hypotheses to test in more detail with other measures. Together with a colleague here at UC Irvine, Marcelo Wood, who is an expert in the molecular mechanisms of learning and memory, we looked at NPAS expression after fear conditioning. So mice were placed in a novel context with a foot shock, and we looked at then the NPAS expression and found that there's certainly a nice regionalization to NPAS expression in the context condition fear learning condition. And we are analyzing this data and think that there's a tremendous promise for this technology for discovering circuits that are engaged in a particular behavior. Okay. So I told you about an instance of neuroinflammation, another instance of neural activity mapping using this brain-wide anatomics. I wanted to now transition to another application for brain-wide mapping, which looks at the impact of coronavirus infection on brain inflammation, specifically on the activation of the brain resident immune cells, microglia. So together with a local collaborator, Tom Lane, we have been looking at how coronavirus induces inflammation in mice. We're starting with the mouse coronavirus, but we'll be working with human SARS-CoV-2, and the coronavirus obviously is broadly known to produce neurological disturbances that are long-lasting. We know something about the general class of coronavirus and their impact on neuroinflammation, but it may well be that the unusual symptoms that patients find after coronavirus infection may be explained by regional vulnerabilities to coronavirus or its neuroinflammatory sequelae. So what we're doing is taking brains from coronavirus infected animals, then labeling the microglia, clearing the brains, imaging them, and then using our quantitative methods to discover regional patterns of neuroinflammation. Presumably this, when tested and validated, could be used to examine whether specific neural agents could reduce the inflammatory impact of coronavirus infection and protect or treat patients. So here is a whole brain that is labeled with IBA1 antibody, which identifies microglia. Here you can see the zoom in, and the microglia here in this case have distributed processes that survey the tissue. We can use our machine learning tools to segment out the data and identify each and every one of these cells across the whole brain and its morphology. And what you can see here in some preliminary data in this work with Tom Lane's lab is that the mouse coronavirus does induce inflammation across the whole brain. And in particular, it seems that certain regions are affected more than others. So in the cortex, in control, you can see that there is less signal in the areas between the cells, but this is enhanced in the mouse coronavirus infected animals. And when we look in the brainstem of the control animals, there's fewer cells than we can see in the brainstem of the coronavirus treated mice. So this is just the very fresh data. We're still looking at this more in depth to try to characterize the regionalized patterns of proliferation and activation of these microglia. And this is just one taste of the kind of quantitative measures that we're employing, showing you here that the IBA1 staining it goes up in conditions which induce neuroinflammation, such as LPS and the mouse coronavirus infection. Also, the area of the cell masks increases in the inflammatory condition induced by LPS or mouse coronavirus. And this makes sense because we know that when microglia become activated, they bunch up and become more amoeboid in form, and that we can now quantitatively detect that using the cell volume mask really gives us an incredibly powerful analytic tool for identifying inflammation by region. Okay, and so we're using this technology not just to look at mouse microglia, but also to look at human microglia, but not in the human brain, instead human-derived, patient-derived microglia that are implanted into a chimeric model. So I've been working closely with two colleagues here at UCI, Matt Blurton-Jones and Rob Spitale, to develop a platform for examining human microglia that are implanted into mice in order to study their biology. And I just wanted to show you a video we made of a cleared mouse brain in which we had engrafted these cells just with a few injections. And what's so remarkable is how widespread the proliferation of these human microglia that are labeled here with a human-specific antibody into the tissue. And in many regions, you get an occupation of the niche which reaches and exceeds 80%. And in fact, we've been able to document that that injections placed in one hemisphere can migrate into the other hemisphere. So this technology also helps us identify and learn about human microglia proliferation, activation, and in this case, chimeric transplantation. Okay, so in the closing minutes, I wanted to tell you about a fourth application that we have for whole brain clearing and quantitation. And in this case, it's about looking at the microglial activation induced by amyloidosis. So here what I'm showing you is the control and experimental activation of IBA1 expression in the 5X FAD aggressive model for amyloidosis. In the 5X animal, of course, there are plaques that begin to form, and we can see in the IBA1 signal the migration and activation of microglia around those plaques. That's easier to see here in this brain cleared image that directly stains the beta amyloid in green and the IBA1 microglial marker in red. And here you can see in the zoom in that the microglia are attacking or congregating with the beta amyloid very closely. So this framework lets us investigate the relationship between microglial activation, like I just showed you, and the deposition of these beta amyloid plaques. I'll show you an image of the beta amyloid plaques right here. You can see this is a whole brain image of those plaques. And in the zoom in, you can see the individual plaques that we again use our computational pipeline to segment out in 3D. We can then analyze these plaques in many different dimensions. What I'll show you here next is in terms of density. So this is the amyloid density across the whole brain. And what you can see is that, that there are regional patterns. So this is just one mouse looking at different planes along the sagittal axis and the Allen reference atlas down below. When we quantify the distribution of this amyloid across different brain areas, you find patterns that you'd expect in the 5x FAD model for amyloidosis. You see preponderance of plaques in the subiculum, moderate levels in retrosplenial, but less so in cerebellum and aspects of the visual system. So this tool becomes a very potent method then to establish the connection between pathogens like beta amyloid and the brain resident immune cells, microglia, to understand their biological link, to investigate potential interventions, and to better understand overall the disease mechanisms, not only in the case of beta amyloid, but presumably in many different experimental models. Okay, so I wanted to acknowledge the individuals who have contributed to this body of work. On the left are members of my lab who are all incredibly hardworking, committed, and joy to work with. Then in the middle, there are many collaborators at UCI and elsewhere. And I wanted to acknowledge the strong support of many organizations to underwrite this work. And so with that, let me end and take any questions you may have. Thank you so much.
Thank you, Dr. Gandhi, for that fascinating and inspiring presentation. We do have a few minutes left for Q&A, and I've compiled a list of questions from the audience. Let me just start with the first. So you've shown some really beautiful images of rodent brains. We have a question that asks, can these approaches to imaging also be used in the spinal cord?
Yeah, in fact, the same clearing technology can be used quite productively in the spinal cord. And if you don't mind, let me just share some slides with you. Okay, so we recently completed a collaboration with Ozzie Stewart's lab to look at the motor system using retro AAV tracing to map the circuitry. So in these experiments, Ozzie's group placed the retro AAV injection at different levels of the cord, and then we cleared and stained and imaged the entire brain and cord together. Let me show you a movie here which lets you see some of the cell bodies of the corticospinal tract that are labeled this way. And what you can see is as we go in, that there are a number of these cell bodies that are distributed through the brain. And Ozzy's lab quantified exactly where these cells are. There, to me, were some very surprising patterns that perhaps, more skilled anatomists of the motor system may be more familiar with, that we found. But we were able to count out the cells that were projecting to different layers, different levels of the cord to better understand the organization of this circuitry. In another project, one of my students has been looking at peripheral nerves and the circuitry of these peripheral nerves interacting with the spinal cord. So in this case, what he's done is extracted and cleared brains with cord intact and the dorsal root ganglia, in which he's used a viral label to infect one of the dorsal roots and get at the cell bodies for these peripheral nerves. And you can see here in the zoom in, in red is the endogenous PV staining, and in green are these labeled peripheral nerve circuits. And the light sheet imaging lets us roam all throughout the tissue and look at the farthest extent of the projections of these cells, lets us zoom in to the dorsal root and even look at features of the axons in great detail. And this is my student Jacob, who's been doing this work. And with the light sheet, we've been able to go in and optically dissect even these dorsal root ganglia and look at fine features all in the intact tissue. And one of the really interesting things that's come from it is that we've been able to not only examine these circuits at the mesoscale perspective, but then also follow up on some of these tissues, looking at them using more traditional confocal microscopy to find more of the microscale information. So together with John Sack and his lab at Davis, we've been looking at the distribution of KV ion channels in these T-junctions here, axonal T-junctions in the dorsal root, and you'll see the axon is labeled in green, and in red in a second you'll see the overlay with the KV channel distribution. And these kinds of studies enable us to go from this micro scale all the way up to the mesoscale that I've been showing over the last few slides. So it's a very powerful toolkit for dissecting spinal and peripheral circuits.
That actually relates to another question that we have. Can you resolve individual connections using these techniques?
You know, there are strong limits to the optical resolution of light sheet microscopy. We generally can't see individual spines as we might wish to. But in the case of projections, we can indeed find some, particularly for sparsely labeled axonal projections, we can use these tools to investigate connections. So here's a story that this slide unfortunately is not updated. This is a published study from my postdoc, Carrie Ha, in which she was examining the structure and function of thalamocortical projections in the visual system. So what she did was she labeled thalamic cell bodies using a viral strategy, and then used multiphoton microscopy to image in awake mice the terminations of these thalamic cells in the cortex. And I'll show you here a movie that zooms in on one such axon, and you'll see it will go firing as the animal. This is a GCamp signal as the animal is watching a movie, and each of these spots that you'll see here, these are individual synapses, and you'll see another sea of synapses from other axons in the surrounding area. So this is the multiphoton imaging. Okay, hopefully you can see that video and the flashing axon. So we wanted to know, okay, well, with the multiphoton, we can see this segment of axon, we can characterize the visual response properties, but can we look at the broader anatomy? So, so Carrie, along with a talented graduate student, Ricardo Azevedo, took the same samples that had been imaged on the multiphoton rig and cleared, stained, and imaged these brains using the light sheet. And so what you can see here is a pseudo-coronal section of that hunk of tissue in the visual cortex, with the binocular visual cortex shown here. And here's the cortical plate, and here's the peel surface. And what you can see is that these individual axons are climbing up and innervating up here in the superficial layers. We were able to segment out an individual axon from these image data sets that we had recorded from during the live imaging session. And we're still investigating the link between anatomy and function, but we already had a hypothesis that some of these synapses would be larger than others. And so the light sheet imaging is allowing us to make that kind of investigation of these individual termination fields. So that's one way in which we're finding that light sheet imaging can indeed be used to look at individual connections.
That's really, really fascinating. We have a rather popular question. Can these approaches be used in other model organisms, such as the rat?
That's a great question. So we have had some success with using rat brains intact in the mesoscale imaging system that we have used to adapt the Z1 light sheet. The rat tissue is, of course, considerably larger, but with some care, one can fit a whole rat brain in the imaging chamber. So on the imaging side, certainly it's quite feasible. One limitation, however, is that the anatomical brain maps that we have for regional demarcation, the annotated brain maps are kind of wanting. So the Allen Reference Atlas is quite detailed and provides for the coordinates for hundreds of distinct brain regions. The maps that are currently available for the rat are not nearly as detailed. So future work needs to be done in order to establish more detailed rat anatomical references. And I'm sure that that will occur in the coming years because of the tremendous benefit that we would have to being able to extend these technologies into the rat model system.
Thanks. Unfortunately, that's about all the time we have left for today. To the audience, if we weren't able to address your questions that you submitted through the chat function, we will reach out to you directly to follow up. I'd like to thank everyone for attending today's webinar and hope you all stay safe, stay healthy, and have a great rest of your day.